Prof Barry Greenberg, who's going to talk about another very innovative study. And just to let our colleagues in the press know, the reason I request people to stand up is we can't see you because the lights are in our eyes. So you're blurred, and that's the reason. So please indulge us if it's possible. Easier to recognize you and invite you to comment. Please, Barry.
Thank you, Salim. I've not edited my slides, and I apologize to the audience for that, so I'll go through them quickly and try to maintain the time frame that Professor Youssef has set here. CUPID-2 was the largest gene transfer study done in cardiovascular medicine to date, and the first one to use a clinical endpoint of morbidity and mortality. The study involved 67 centers around the United States, Europe, and in Israel, and these are the committees that helped run the study. The background for the study is that SERCA2a, an enzyme that sits on the endoplasmic reticulum of cardiomyocytes, plays a critical role in the regulation of calcium homeostasis within the heart. In the failing heart, it's known that there's a deficiency in SERCA2a activity, and that correction of this leads to recovery of calcium homeostasis as well as normalization of individual myocyte function. And this is data from isolated myocytes from the failing human heart. Earlier studies in animal models showed favorable effects, and these were replicated in a small pilot study called CUPID-1, in which AAV1 SERCA2a was delivered to patients with advanced heart failure, and there were a variety of positive results that were seen. CUPID-2 was then organized to replicate the findings found in CUPID-1. The inclusion criteria included patients with low ejection fraction, symptomatic heart failure of either ischemic or non-ischemic etiology. AAV1 SERCA2a was administered intravenously through the coronary arteries with 10-minute infusions at only one single time point. The primary efficacy endpoint was recurrent hospitalizations for heart failure in the face of terminal events. Secondary endpoint was time to the first terminal event. The sample size calculations indicated that we would need 250 patients, and all of them, after administration of AAV1 SERCA2a, were followed for at least 12 months. This was an event-powered study, but the events were reached within the 12 months, but all patients were followed for 12 months, so we actually had an excess of events in the study. These are the patients who were included in the clinical trial, and basically they had evidence of moderate to severe heart failure, no difference between the two study groups, and they were well treated. The primary efficacy endpoint results are shown here. There were 128 of these in the placebo and 104 in the AAV1 SERCA2a. And thus this did not achieve statistical significance. The hazard ratio was 0.93, a 7% risk reduction. Here are the Kaplan-Meier curves demonstrating that. The secondary efficacy endpoint, time to first terminal event, also failed to show a significant effect of AAV1 SERCA2a. When we did subgroup analysis, there was no indication from either predefined or exploratory subgroups that there was a favorable effect of this therapy. And for exploratory efficacy endpoints, also no significant effect of AAV1 SERCA2a. Safety, there were no safety signals, and I won't go through this at any length. So in summary, treatment of patients with moderate to severe heart failure with AAV1 SERCA2a this treatment did not significantly reduce the likelihood of either recurrent hospitalizations or terminal events. There was no evidence of improvements for these endpoints in any predefined or exploratory subgroup, and there were no beneficial effects on exploratory efficacy endpoints. There were no safety issues that emerged during the course of the study. Thus, our main conclusion is that AAV1 circuit 2A failed to improve the clinical course of patients with moderate to severe heart failure and reduced ejection fraction on top of background therapy. This is an important study for a number of reasons. Gene therapy is a very intriguing approach for treating a variety of cardiovascular disease, and this was the largest study done to date. I think it's vitally important that we understand why this study did not work out, and there are a number of questions that one might ask, including those related to study design. A fundamental question would be whether or not approaching a single abnormality within cardiomyocytes can rescue the heart failure phenotype. And then finally, questions related to adequate drug delivery in a very complex clinical milieu. All of these are currently being looked at, and hopefully this will stand as a building block for further therapies in the future. Thank you.
Well, thank you. Questions from the floor. Thank you, Barry. This is, again, a very important, a challenging study, if I may add, these kinds of studies. So questions from the floor, please. Well, while you're thinking about this, oh, sorry, is there a question? Oh, there are. Again, please, it's hard for me to see people from here. The lady in front, and then, the gentleman at the back. Is that Larry Houston? Yeah.
Larry Houston, Cardiobrief.
Right.
I just want to know, is there the cycle with gene therapy, and now it seems maybe to be the same with stem cell therapy? There's been so much earlier hype and so much disappointment. Do you come away from this with anything, with genuine hope for the future, or you think it's just a wrong direction? And I wonder if any of the panel members would want to comment on that.
No, I think, again, if you look at development of any novel approach for treating a common disease like this, it's not always a smooth road, nor is it a quick one. And I think to look for the definitive result in a single study like this is probably being over-optimistic. We did show evidence of sustained transgene expression within the myocardium of these patients, and I think that's an important finding. I didn't present that data today, but there certainly was that information available. I think there are a number of issues that did come up about delivery of the drug in this patient population that need to be closely examined, and I think we'll hear more about gene therapy in the future.
Yeah, I think that's an excellent question. I think people should take away from this two things. One is, without these trials, we don't know whether a given hypothesis is right or wrong. But even this trial, which is neutral, doesn't mean the hypothesis is wrong. It could be the delivery system needs tweaking, the dosing needs tweaking. So there's a lot to learn, but Larry is right. I think the expectation that we will see magic isn't happening. I think there was a question up here. Okay, any other questions for Barry? I mean, I think Larry Houston asked a good question. You know, stem cell research has been around for 30, 40 years. And other than in bone marrow transplantation, we have, and say maybe the eye. There are studies with the eye where vision has improved. Yeah, we certainly haven't seen much in cardiovascular disease, and there have been lots of little trials, and yours is one of the largest trials. Do you think enthusiasm will wane, which would be a mistake? Do you think people continue to indiscriminately be excited without the data, which would also be a mistake? So what would you think would be a rational middle path?
So I think a rational middle path would be to go back to the fundamental biology of delivery of these new therapies in the very complex milieu of patients with advanced heart failure who have a variety of different systems activated internally. There are also environmental effects, including presence of neutralizing antibodies that can affect the efficacy of gene therapy, and also the therapeutic agents that are currently being used. These are all very different from what we're seeing in the animal models, where there was clear-cut evidence that you could improve the lifetime of animals given experimental heart failure. Clearly, human patients are different, they're more complex, and I think moving away from this would be a mistake at this point, but I think that having undue expectations would also be a mistake, as you indicated, Salim.
Well, thank you, Barry. We've really done very well.